For Chapter 8, Segment 3, which is the pages 246 to 253, starts with the section titled Plasticity in Cortical Maps, which means receptor fields can be changed by experience. These cortical maps can change with experience, and the one example that they gave was that when a finger is removed for whatever reason, the map changes so the other fingers overtake the missing finger's place on the map. Next section is pain is an unpleasant but adaptive experience. Pain is, by definition, the discomfort that's normally associated with tissue damage, and the disease that people are born without the ability to perceive pain is called congenital insensitivity to pain. And from the study of this, it's given us insight to the benefits of pain. It guides adaptive behavior by signaling harm to our bodies, and it gives us lessons on how to avoid injury. 3 ways that pain can help us is through short-lasting pain, which causes us to withdraw from a source, usually the reflex, and prevent further damage. The second being long-lasting pain, which provides behavior that promotes recuperation, including sleep, feeding, drinking, and stuff of that nature. The last being the expression of pain, which is usually a social signal to other animals to avoid a certain behavior, or the pain that we are experiencing now will also be experienced by them. The next section is human pain can be measured. Learning, experience, emotion, and culture all affect the perception of pain, but there is a McGill pain questionnaire, which is a list of words ranged into classes that describe 3 different aspects of pain, these being sensory discriminative quality, the motivational affective quality, and the cognitive evaluative quality of the pain. Different types of pain can be described using these 3 different words, and you can usually distinguish between different types of pain in different areas based on the selection of words. So example, tooth pain would have different chosen descriptors for each of these categories than for foot pain. The next section is peripheral receptors get the initial message. In most cases, the initial stimulus for pain is the destruction or injury of tissue adjacent to certain nerve fibers, and damaged tissues usually release chemicals that activate pain fibers within the skin. Nociceptors themselves are receptors that respond to stimuli that produce tissue damage or pose threat of damage. Free nerve endings are axons that terminate in the skin without any special cell associated with it, and that detects pain and or changes in temperature. They have special receptor proteins on the cell membrane that respond to various signals, and different nerve endings have different receptor proteins, so overall they report different stimulation. Next section is titled Hot, Cool, Cold, or Cool. The discovery of the transient receptor potential vanilloid type 1 receptor, which is TRPV1 for short, comes from the study of capsaicin, which is a compound synthesized by various plants to deter predators by mimicking the experience of burning. This is usually found in like spicy peppers. The receptors that respond to this spice normally respond to a sharp increase in temperature, and those being what we said before, TRPV1, which is a receptor that binds to capsaicin to transmit the burning sensation from chili peppers and normally detects a sudden increase in temperature. So the study of capsaicin revealed the specific receptor that responds to a sudden increase in temperatures. These TRPV1 receptors use C fibers, which are small unmyelinated axons that conduct pain information slowly and adapt slowly, and are responsible for second wave pain, which is the dull, lasting ache. The other pain receptor or temperature receptor that was found, which is related to TRPV1, is the transient receptor potential type M3, which is a receptor found in subnerve endings that opens a channel in response to an increase in temperature. The difference between TRPM3 and TRPV1 is that TRPM3 detects higher temperatures and it doesn't respond to capsaicin. The receptors of TRPM3 PM3 are found on nerve fibers that are longer than those for TRPV1. So they're on long axons called A delta fibers, which are moderately large myelinated fast-conducting axons transmitting pain information, reporting to the spinal cord, and they're responsible for first wave pain. So TRPM3 and TRPV1 are responsible for the detection of sharp increases in temperature. TRPM3 is the first wave, TRPV1 is second wave because it's slower conducting, TRPM3 is faster conducting. Cool menthol receptor 1, or CMR1, is a sensory receptor found on some nerve endings that opens an ion channel in response to a mild temperature drop or exposure to menthol or cool temperature, which are found on small C fibers and therefore are slow acting. The receptor for itch responds to histamine released from mast cells in the skin, or others don't respond to antihistamines at all. Both use TRPV1, which is, again, slow, uses slow conducting C fibers to the spinal cord. And natriuretic polypeptide B, or NPPB, is a peptide neurotransmitter used by neurons reporting itch to the spinal cord. The neurons in the dorsal horn are specifically what they're reporting the peptide neurotransmitter to. When you scratch the affected area, there's temporary relief by silencing spinal neurons that respond to NPB fibers and report itch sensation to the brain. Spinal mechanisms to suppress the response to itch signals use NPY for the transmitter. The next section is the pain receptor. Overall, it's unclear which chemical is crucial for activating nociceptors because there's so many chemicals that are released. But once nociceptors are activated, they use a particular type of voltage-gated sodium channels to produce action potentials to report pain to the brain. NAV1.7, or SC9A, is a voltage-gated sodium channel almost exclusively used by nociceptors to initiate action potentials, and this is expressed in many free nerve endings. So SCN9A is the specific voltage-gated channel that is used to produce action potentials to report pain to the brain. Next section is spinal pathways transmit pain information. Within the central nervous system, there are special pathways that mediate pain and temperature information. These are anterolateral or spinothalamic system. The definition being a somatosensory system that carries most of the pain and temperature information from the body to the brain. Free nerve endings in the skin send axons to synapse on neurons in the dorsal horn of the spinal cord. These spinal cord neurons send axons across the midline to opposite sides and up the anterolateral column of the spinal cord to the thalamus. And then the spinothalamic system, pain information crosses midline in the spinal cord before sending to the brain. So these two have different methods for sending pain information. And afferent fibers from the periphery that carry nociceptive information probably use glutamate to excite cells in the dorsal horn and release neuromodulator substance P, which is a neuropeptide, in the dorsal horn. Postsynaptic cells then take up and remodel their dendrites due to the neuromodulator. And that is segment 3 of chapter 8.